Hello everyone and welcome to Lecture 5. We're going to talk about synaptic transmission, starting with the synthesis of neurotransmitters, their release, and then ending with postsynaptic signaling. Okay. Neurotransmitters are the chemical messages that will be sent between the neuron and its target cell. We're going to create neurotransmitters with specific enzymes depending on the neurotransmitter, either in the soma, in the case of peptide transmitters, or at the synapse itself, which is the case with your classical neurotransmitters. Now, not every synapse uses neurotransmitters. Okay, a very small minority of synapses in the nervous system are what we call electrical synapses, and these are gap junctions between two nerve cells that allow ions to directly flow from one cell to the other, so that electrical activity that's generated in the presynaptic cell moves directly to the postsynaptic cell. And that's what we see on the left. On the right, we have a chemical synapse. And this is the overwhelming majority of the synapses in our nervous system. You have a clear separation between your presynaptic and postsynaptic cell. There's going to be the release of a chemical transmitter that diffuses across this space, binds to receptors, and then generates a new electrical event. Okay, so we're not directly passing the electrical activity from our presynaptic cell to the postsynaptic cell. We translate the electrical activity into a chemical signal, which then gets translated back into electrical activity at the postsynaptic cell. Now, the good thing about the electrical synapses is that they allow for very fast charge transfer, okay, because there's direct flow through those gap junctions. What we're looking at here would be the gap junction. This is created by proteins called connexins, which join up with connexins on the other cell to form these pores. Unpaired connexins form what we call hemichannels, and they just essentially leak from the cell. But by joining up the two connexins from one cell and another, we create a direct pathway that allows the two cells to share their cytoplasm and thus their electrical activity as well. Now, for the most part, these are really only excitatory. If we generate an action potential that moves down the axon, that can never become an inhibitory signal. It can only excite, right? Because what we're doing is we're generating depolarization in our presynaptic neuron, and that depolarization flows directly from the presynaptic to postsynaptic neuron. You'll notice that this is very, very fast, though. And so the good thing about electrical synapses is that it allows your cells that are connected with them to essentially form one large network that all acts together. You can see there's a very brief delay at this synapse. That's because we're not relying on vesicular fusion, diffusion of neurotransmitter, binding to the receptor opening. All that takes a little bit of time. Here, instead, all we're doing is having the electrical signal flow directly from one cell to the other. So in pacemaking networks or collections of neurons that all fire synchronously, there's where you find your electrical synapses. And these are indeed the same gap junctions that also join our astrocytes together into their networks. But for the most part, we're going to be dealing with neurotransmitters, and so it's a good place to start with what a neurotransmitter is anyway, since the definition has evolved as we've discovered new different neurotransmitters. The three criteria that seem pretty good to me would be that they have to be synthesized in neurons, so neurons have to create this substance. The neuron has to release this substance in an activity-dependent manner, and the released substance then needs to bind to nearby cells and cause some sort of reliable change. Conventionally, what we're talking about here is that we're going to have the presynaptic release of a neurotransmitter. It's going to bind to postsynaptic receptors, and then we're going to clean up that neurotransmitter through a couple different ways. And that's what we can see summarized here. All right, we're going to go through all these steps. It's going to start with calcium influx. That's number one. Number two, that's going to drive the release of neurotransmitters. They will bind to their receptors, either inotropic, 3, or metabotropic, 4. They can then be enzymatically degraded, shown in 5, or taken back into the cell, as shown in 6, or they, if they aren't, will diffuse outside of the synapse, as shown in 7, where they may be picked up by glial cells, as shown in 8. If we're fusing a bunch of vesicles, we have to then recycle our vesicles. That's what's shown in 9 here. And if we have a very high level of activity, we can actually release peptide transmitters that are stored in those dense core vesicles, shown in 10, but from a different site. And that's what's shown in 11 there. So there's a lot going on in this image, and it's all pretty accurate. We'll go through those steps. Okay. So we're going to have to synthesize our neurotransmitters. That's the first step. You got to make them if you're going to release them. And we're going to do this in 2 different areas depending on the type of neurotransmitter. Your classical neurotransmitters are going to be synthesized at the synapse. So what we do to make our classical neurotransmitters, which are shown on the left there, we synthesize the enzymes in the cell body, because that's where our nucleus is, so that's where gene expression takes place. Once we create the enzymes necessary to make our classical neurotransmitter, we then transport those down the axon in what we call slow axonal transport. Those enzymes eventually reach the presynaptic terminal, and there they will generate the neurotransmitter locally. This is good because it allows for recycling of the classical neurotransmitters. Because we're using classical neurotransmitters like glutamate, GABA, all day, every day. Peptide transmitters, on the other hand, we don't release as often because they're a little more costly to produce. Peptides are just 2 or more amino acids strung together. We synthesize these in the cell body because, again, we're putting together amino acids. These are essentially little proteins. That has to happen at the cell body. We then transport the peptide transmitters down the axon by fast axonal transport and release them from the dense core vesicles. We don't recycle our peptide transmitters though, so we have to recreate these brand new every time we need them. So they're a little more costly to produce, so we tend not to release them as often as we do classical neurotransmitters. It requires much stronger activity to release a peptide transmitter. Okay, so at the synapse, once our enzymes have made it and they start making this transmitter, what we have to do is then get that transmitter into a vesicle. Classical neurotransmitters are all packaged into vesicles. Now, it would make the most sense to simply put a transporter protein in the vesicle and have that use ATP to move the neurotransmitter into the vesicle, but that's not what we do. That's not how nature has set this system up. Every vesicle is equipped with a proton pump instead. This is the enzyme that breaks down ATP to generate a proton gradient. It moves protons into the vesicle. All right? And since protons are a measurement of acid, that means we are acidifying the vesicle. We're putting a whole bunch of protons inside this vesicle, so it has a very high concentration. That means it wants to, by diffusion, leave the vesicle. So that's the energy that we're using then to move our neurotransmitter, which is shown as a little red circle in this cartoon. We move that into the vesicle by allowing the protons to leave. So it's a little more complicated than you might think it needs to be, but this is great because we use the same proton pump for every neurotransmitter. So it's the same protein that's hydrolyzing ATP to generate that proton gradient. And then we just simply need passive transport of our neurotransmitters. Either way, it accomplishes the same task. We package our neurotransmitters into vesicles. And because neurotransmitters are packaged in vesicles, that makes the signals that they generate very reliable. It's what we call quantal. So at a given synapse, you're going to get the same reliable result, the same reliable response to each vesicle, and that's because all the vesicles are about the same size. They have about a 50 nanometer diameter, and they get filled to about the same concentration of neurotransmitter, which means that we have about the same number of neurotransmitters in every vesicle. So if you were to release one vesicle or another, it doesn't matter. You're releasing the same amount of neurotransmitter. And that's why we say that signaling is quantal. If you record the spontaneous activity at a synapse where you're looking at the release of one single vesicle, you see about the same reliable postsynaptic response. If you were to get two vesicles, it becomes about twice that amount, 2 quanta, as opposed to 1. Now, of course, depending on which synapse we're at, a single vesicle might produce a different postsynaptic potential, so they might have different quantal size. If a synapse is closer, it could, in theory, produce a greater postsynaptic potential than a very distant synapse. And the number of vesicles that are going to be released with each action potential, that can also vary from synapse to synapse. And that's what we call the quantal content. Some synapses have a very low release probability, and they'll release anywhere from 0 to 2 vesicles. Others might release upwards of 6 vesicles, depending, of course, on the release probability. So if you were to measure postsynaptic activity, which they've done, on top they're looking at postsynaptic activity with different concentrations of calcium. By having more or less calcium around, you're going to make neurotransmitter release more or less likely. And so you can see with higher levels of calcium, shown on the bottom versus the top, there's a lot more postsynaptic responses. If you were to then quantify the size of these postsynaptic responses, that's what we're looking at on the bottom, you'd find that they tend to be multiples of some basic response. In this case, about 0.4 millivolts would be 1 quantum. So that's about the response you get from one vesicle. You see another peak at about 0.8 millivolts, another one at 1.2, so that's the response to 2 vesicles, 3 vesicles. And there are a variety of reasons that this doesn't sum perfectly. Again, differences at different synapses, so differences in quantal size and quantal content can affect this. But you do tend to see this quantal release because of vesicular packaging, because we tend to release the same amount of neurotransmitter at each synapse. Because they have a given quantal size and quantal content, we get these reliable responses. Okay, the action potential is really only important because it causes the release of neurotransmitter. And it does that by allowing the influx of calcium. Calcium then affects the interaction between snare proteins and drives vesicular fusion. Okay, so it's all going to start with the action potential. We should understand the action potential by now, so we're not going to review that. We should know that a wave of depolarization is going to hit the presynaptic terminal, and when it does, that's going to activate voltage-gated ion channels. In this case, not voltage-gated sodium or potassium, but instead voltage-gated calcium channels. So the depolarization causes voltage-gated calcium channels to open. That's step 1 in this cartoon shown on the right. When the voltage-gated calcium channels open, calcium rushes in. And we know that calcium is absolutely required for vesicular fusion. If we were to remove calcium, that's what we've done here, we can see that we get no further excitatory postsynaptic currents. So on the bottom, we're looking at mini excitatory postsynaptic currents. Those aren't driven by calcium, and that's the response to one single vesicle. This is just spontaneous release, and you can see we're getting somewhere on the order of 20 picoamps, so much smaller than the whopping 10 nanoamp response for the excitatory postsynaptic current. Okay, the solid line on top is showing you the postsynaptic response in the presence of calcium. The dotted line is showing you the postsynaptic response whenever we have cadmium. present. That's a voltage-gated calcium channel blocker. So when we block our voltage-gated calcium channels, no more excitatory postsynaptic current. On the other hand, if we were to directly stimulate calcium increase in the absence of action potentials, we can induce vesicular release of neurotransmitter. And that's what they've done here. This is what we call calcium uncaging. They have calcium wrapped in this chemical cage that's light sensitive. So when you shine a light, you cause the release of calcium. And that's what we're looking at in the top portion of this. Delta F over F is showing you the amount of fluorescence change, which is a readout of the amount of calcium being released. So you can see the thin solid line, the thick solid line, and the dotted lines are going from the least to most fluorescence, indicating the least to most calcium, and that's estimated on the second line. We don't really care about that. We care about the bottom response. So the thin line, the one where we have the least amount of calcium, we get a very small postsynaptic current. The thick line, we have more calcium, we have a greater postsynaptic current. And the dotted line where we have the most calcium release, that's where we have the greatest postsynaptic current. That's because we have the greatest amount of neurotransmitter release. Now in this case, whenever we're doing calcium uncaging, we're releasing calcium all over the presynaptic terminal, but that's not what really happens whenever we get an action potential. Instead, we only get calcium increases immediately surrounding our voltage-gated calcium channels, in what we call calcium microdomains. So within about 0.1 micron radius, we're going to have about 1,000-fold increase in calcium. We're going to be in the hundreds of micromolar concentrations. This is well above what we need to stimulate release. So the active zone, the area where we release neurotransmitters, is going to be littered with about 100 voltage-gated calcium channels. So when the action potential comes down, most, probably not all of these, but most will be activated, so we'll get substantial calcium increase. And in the regions immediately surrounding those calcium channels, we're going to have a substantial increase in calcium and create what we call those calcium microdomains. Alright, now the reason that we only create the microdomains is because calcium is an important secondary messenger, and we want to make sure that we only increase calcium at the sites where we need it to be increased. And that's really only at the active zone there. So only at the place where we have our vesicles docked and ready do we also have our voltage-gated calcium channels. Alright, we wouldn't want to increase calcium just throughout the entire cell, because then calcium would interfere with the normal function of other proteins. So it's very, very local. And in fact, we quickly clean up that increase in intracellular calcium. We only get about a 10% increase in the basal levels of calcium following an action potential. And then these are quickly reduced back to normal levels. Because in that presynaptic terminal, we still have mitochondria, and mitochondria buffer calcium. So does the smooth endoplasmic reticulum that's present. And what this does is prevent calcium from building up, so that every time we fire an action potential, we're starting off with the same very low 0.1 micromolar levels of calcium. So this way, each action potential gives you the same reliable release of neurotransmitter, right? Because we don't just keep elevating our intracellular calcium levels. This would eventually kill the neuron. So we keep everything very tightly wrapped up in the active zone. We're only increasing calcium immediately surrounding voltage-gated calcium channels. Anywhere outside of that, it only has about a 10% increase, which isn't enough to do anything. Then that's quickly buffered by mitochondria and smooth ER. Now, whenever we're talking about vesicles, we should keep in mind that we have really two pools of synaptic vesicles. We have the readily releasable pool, which is down in the active zone. These are docked and ready. They are waiting for that calcium increase to drive their fusion with the membrane. So the readily releasable pool is a small collection of vesicles. In our central nervous system, there's usually only about 6 or 7 vesicles in the readily releasable pool. The rest of the vesicles are going to be found in the reserve pool. So a few hundred nanometers away from that active zone, we have a big collection of vesicles. They're filled with neurotransmitters. They just haven't made their way down to the active zone yet. So they're just kind of sitting. They're the savings account of the presynaptic terminal. Once we have vesicular fusion and some vesicles are removed from the readily releasable pool, then our vesicles move on down to the active zone. Now the trafficking of vesicles is of course controlled by proteins, as everything is in life. The protein that probably controls this would be synapsin. Shuttling from the reserve to readily releasable pool is still somewhat of a mystery, but what makes the most sense is that synapsin, which is this peripheral protein found on our synaptic vesicles, so it's attached to the vesicle, but it's hanging off. It can also attach to the cytoskeleton in the presynaptic terminal and hold the vesicles in place. Once we get our calcium increase, that's going to not only drive the fusion of our readily releasable pool, that's also going to lead to the phosphorylation of synapsin. So the calcium influx is going to activate some enzymes that then modify synapsin and cause it to let go of the cytoskeleton. So calcium is not only driving vesicle fusion, it's also leading to the modification of synapsin that allows the vesicle to float on down from the reserve pool into the readily releasable pool. So this allows our readily releasable pool to be replenished in an activity-dependent manner. Kind of nifty stuff. Now the actual fusion of vesicles is carried out by the snare complex. We're going to walk through these steps a couple of times in just a moment, but let's familiarize ourselves with the players. The snare complex is this collection of proteins. There's a few different ones. They wrap around each other. It contains 4 alpha helices. An alpha helix is just a basic unit of secondary structure that proteins adopt. So it's a helix. It's kind of like a little spring, and there's 4 of those kind of wrapped around each other. One alpha helix is going to come from the snare protein on vesicles. That would be VAMP or synaptobrevin. So the vesicle itself has 1 snare protein that gives 1 alpha helix. The presynaptic membrane at the active zone also has a snare protein embedded in it. That'd be the target snare. That'd be syntaxin. This also contributes 1 alpha helix to the snare complex. The other is the other part of the protein is embedded in the membrane. And then there's SNAP25. This is the final snare protein. This is not embedded in any membrane. Instead, it just kind of contributes 2 additional alpha helices to hold the snare complex together. And then there's this important accessory protein called MUNC18 that kind of holds the snare complex in a loose conformation and only allows it to tightly interact once we have a calcium increase. And more on that in a moment. Let's review why snare proteins matter. We think that snare proteins are absolutely critical for vesicle fusion because there are toxins that actually target and break down snare proteins. And when these toxins, when neurons are exposed to these toxins, they can no longer carry out synaptic transmission. They can no longer fuse their vesicles with the protein. So the clostridial toxins are these neurotoxins made by the genus Clostridia. So this is just a genus of bacteria. And there's botulinum toxin, there's tetanus toxins. They're going to cut different snare proteins. And you can see in this cartoon which one they're going to target. For example, botulinum toxin C1 is going to cut up SNAP25 and syntaxin, whereas botulinum toxin B is going to cut up synaptobrevin. Tetanus toxin will also cut that. So whenever we introduce these clostridial toxins, like tetanus toxin, we get a buildup of synaptic vesicles, and that's what we can see here. The control without tetanus toxin is shown on the left. We can see that there's 3, maybe 4 docked vesicles at the active zone. And of course, the synapse shown on the right, where we have tetanus toxin, the synapse is enlarged in size. It's because of the accumulation here of at least a dozen vesicles. So the vesicles can make it there, they dock, but they can never bind. They never fuse anymore. And so this is why botulinum toxin and tetanus toxins will cause paralysis, because our nerve cells can no longer communicate with their targets. They can no longer fuse their vesicles. No matter how much calcium they have, it doesn't matter. The snare proteins are broken. Okay, what are those snare proteins doing? We don't know. There are two likely explanations. It could be any combination of one or both of these. The first potential mechanism for vesicular release would be that synaptotagmin is going to insert into the membrane. Okay, synaptotagmin is this calcium-binding protein that's going to be linked to our synaptic vesicles. Okay, it's not synaptobrevin. Okay, it's not the snare protein. This is a different one. This is synaptotagmin. This is the calcium sensor. Okay, we're going to go through this entire illustration a couple of times. So we start off here. So our snare complex is going to kind of form at the presynaptic site. We have syntaxin, SNAP25, and MUNC18 all hanging out together. We're going to dock and form this snare complex. All right, so let's see that. Our vesicle there with synaptobrevin and synaptotagmin is going to come on down. What I want you to do is follow this illustration to the right. We're going to look at the right of this first, and then we'll go through and walk down the left-hand side. Okay, so in this model, our synaptobrevin is going to associate loosely with the snare complex, and you can see here that synaptotagmin is associating with the plasma membrane. All right, once calcium enters, what this is going to do is, for one, it's going to allow the snare complex to form very tightly. MUNC18 gets out the way, but synaptotagmin, you can see, is going to pull that vesicle close into the membrane, and it's going to drive fusion. So what we think is going on is that synaptotagmin, whenever it's bound with calcium, can actually insert into the plasma membrane, and it can disturb that lipid bilayer. And by disturbing that lipid bilayer, what it does is make it much, much easier for a vesicle to then insert its membrane and fuse, because we've disrupted the strong stability of the plasma membrane by just opening it up with synaptotagmin. So it essentially functions as kind of like a drill that goes into the membrane. All right, and that's what we see here on the right. Synaptotagmin directly inserts into the membrane and kind of forces a hole to form that then allows vesicular fusion. All right, the other possibility is that synaptotagmin is going to, instead of inserting into the membrane, it's going to allow that snare complex to form. So when we don't have any calcium around, our snare complex is held together. It's in this kind of loose state, and there's a new player that we have to keep in mind here called complexin. This is another protein that kind of inserts into the snare complex and prevents it from interacting very tightly. So now let's follow this illustration to the left, all right, rather than going to the right. You'll see on the left there, now synaptotagmin is not inserting into the membrane. Instead, it's associating with that snare complex. Okay, so complexin comes in, and it's kind of preventing the snare from zippering up freely. Whenever we have calcium influx following an action potential, what synaptotagmin does when it binds calcium is actually then interact with the snare complex and displace both MUNC18 and complexin. When it removes MUNC18 and complexin, the snare complex zippers up very tightly. Those intermolecular interactions release a sufficient degree of free energy to drive vesicular fusion. So we don't need to poke a hole in the membrane as in the first model. In this case, all we need to do is allow our snare complex to zipper up tightly, and that will give us enough energy to drive vesicular fusion. So these are two possibilities. whenever we're dealing with very small molecular mechanisms like this, it can be difficult to tease them apart because this happens so quickly. Either one of these are plausible at this point. We haven't ruled one or the other out. Because vesicular fusion is very, very fast. And in fact, the way that neurotransmitters first leave the cell is somewhat open to debate. Because it's going to take a couple hundred microseconds for vesicular fusion to take place after the action potential arrives. So the fusion pore that forms could either be protein lined or it could be instead just the lipid line protein pore. We're not sure if there's this transient protein lined intermediate. So the snare proteins may actually form a pore through which neurotransmitters will transiently escape the presynaptic terminal. Whether or not this is true is still debatable. So what we should understand with snare proteins is that they are responsible for driving vesicular fusion when they interact with one another. And that interaction is, of course, controlled by synaptotagmin and calcium. But just keep in mind, it's not just that simple. Okay, the snare proteins might actually be doing other things besides just allowing the membrane of the vesicle to fuse with the membrane of the presynaptic terminal. But in the end, that's what happens. Vesicles fuse with the presynaptic terminal. Now, let's do a little bit of a reality check here. Your typical presynaptic terminal contains about 270 vesicles. If you only release 1 or 2 in a given action potential, it's going to take less than 200 of those to completely deplete the synapse. If you fire 10 action potentials per second, you got about 20 seconds to live until you're out of vesicles. Of course, vesicle recycling is a very important part of neurotransmission. If we didn't recycle our vesicles, we'd run out, we'd be paralyzed, we'd stop breathing, and life would end. Of course, that's not the case. When vesicles fuse, at the same time, the presynaptic terminal's creating new vesicles. So it's recycling that membrane. But in order to do that, it has to break apart those proteins that we just put together. So we went through all the trouble of having calcium come in, drive the snare complex formation, and then cause neurotransmitter release. That's what we see happening on the top of this illustration and then going down to the bottom right. We have neurotransmitter release. But this snare complex is very, very stable. So if we're going to break it apart so that we get our vesicular snare, VAMP, or synaptobrevin, separated from our target snares, we're going to have to spend some energy. And that's what NSF. That's what NSF is going to do. NSF is an enzyme that uses ATP to break apart the snare complex. And that allows us then to recycle vesicular proteins without taking away the proteins that should be embedded in the presynaptic membrane. So NSF comes in, breaks apart the snare proteins. The vesicular snares then actually diffuse outside of the active zone and then get taken up. Here we can see vesicular recycling with electron micrographs. So here if we just look at the top row, we got A, B, C, and D. We're looking across time, immediately before and then after an electrical stimulation. So after electrical stimulation occurs, once we have our action potential take place, what you can see is that outside of the active zone, outside of that postsynaptic density there, they start to pull off a vesicle. All right, and they've highlighted that with the arrow. This starts to pull inward in part B. In part C, you can see it starting to pinch off the vesicle, and then in D, it has recycled a bit of that membrane. And this allows us to then replenish our presynaptic terminal with vesicles. Now, once we get our proteins outside of the active zone, we can then start to pull them up into vesicles, and this is accomplished by adapter proteins. These are just proteins that will bind to the vesicular proteins like synapsin or VAMP, and then create a binding site for the protein clathrin, so that we can then create our clathrin coat and pull up the vesicle. The adapter proteins are there so that clathrin doesn't have to try to identify all the different vesicular proteins. That's what the adapter proteins do. The adapter proteins are the ones that go hunt out the vesicular proteins, and then they mark the region that clathrin needs to bind. Clathrin is yet another protein, of course, like pretty much everything we're going to talk about. Clathrin is going to bind to those adapter proteins, and it's going to create this cage, soccer ball looking structure. As it's building the cage, it's creating that little bubble. It's creating the invagination of membrane. This is going to be further shaped by other accessory proteins, the bar proteins, to make sure that we're pulling up a nice little sphere of membrane, and then we pinch it off using dynamin. This is a protein that hydrolyzes GTP rather than ATP, and it's essentially a coil that when it hydrolyzes GTP, it elongates, becomes thin, and essentially just pinches off that little vesicle. Once the vesicle is removed from the presynaptic membrane, clathrin dissolves and then can go and reform to make a new vesicle. And then once we've released our neurotransmitter, of course, we're going to have to create some sort of postsynaptic response. These can be either electrical, chemical, or both. Now, synaptic currents can have two components. There can be a fast and a slow component. The fast components are caused by ionotropic receptors, so ligand-gated ion channels, and then there's a slow metabotropic component. That's caused by G-protein coupled receptors. So they've marked the early and late components here. If you look in panel A, what they're showing you is the response of the early and late component across different membrane potentials. So if you look at negative 66 millivolts, we get an inward current in both cases. At negative 78 millivolts, we really don't see any response at all at the early phase. The late is still coming inward. At minus 102, we get an outward current with the early response, and really nothing with the late. And at negative 115, they're both outward. Now, the whole purpose of doing this is to show that there's two different ions mediating this. And so if we look over at part 2, they've plotted the relationship between the postsynaptic response and the membrane potential. So membrane potential is shown on the x-axis and the postsynaptic response is on the y. So you can see that the early response reverses somewhere around minus 78, something close to the reversal potential of chloride. Thus the early component is a chloride channel, clearly. The late component reverses around negative 100 or so, which is very, very close to potassium's reversal potential. So as such, what we see here in those recordings, we see an early chloride conductance and then a later potassium conductance. Both of them are inhibitory, but they're going to be caused by two different receptors. The fast component's caused by ionotropic receptors, so just a chloride channel that opens up and immediately allows chloride to flow into the cell. The late component is caused by metabotropic receptors, where the neurotransmitter binds, but not to an ion channel, it binds to a G-protein coupled receptor. When it does this, the G-protein inside the cell gets activated, and it then goes and interacts with its target proteins. The target protein then, either directly or very much indirectly, causes the ion channel to open up. So it's a little bit slower because you have a few middlemen. You have the G protein, you have the target of the G protein, you have the targets of the target. And that's why you see the difference in timing. That's why that late component is coming on almost 100 milliseconds later. That's because of the intracellular signaling that had to occur to activate that potassium channel. Okay, ionotropic receptors are really just ion channels. That's it. The neurotransmitter is going to bind to the channel, and then that's going to cause the pore to open. So it's not gated by the membrane potential. It's not voltage gated. Instead, it's ligand gated. When the ligand binds, it opens. The movement of this ion, or the ionic current, is going to move the membrane potential toward the reversal potential of that ion. So if we open up a sodium channel, we're going to depolarize the membrane potential. If we open up a potassium channel, we're going to hyperpolarize. What we can see here is the probabilistic opening of ion channels. So the experimental setup is shown in the top. What they've done is patch clamp recording, in this case outside out, so that the acetylcholine binding site is exposed. They add a little bit of acetylcholine. It binds to the receptor. It opens. Sodium flows into the pipette, and you can detect that current below. So shown below in red with a yellow background, we can see when the channel is open, we get the inward current of sodium. When it's closed, we don't. And even though acetylcholine is always there, the channel is not always open. Okay, it's probabilistic. And this is exactly how it works in real life. When acetylcholine is released, acetylcholine receptors don't all open immediately and stay open until the acetylcholine is gone. They just become far more likely to open. But of course, at a given synapse, you're going to have not just one channel, you're going to have thousands of channels potentially. And so by summing together all of those channels, you don't get the square wave anymore. Now what you get is a probability based smooth response. So you can see when you compare a single channel versus a few channels versus all the channels at the synapse, it goes from digital, all or none, to just kind of a smooth response. And of course, the number of channels that opens up will directly relate to the amount of membrane potential change that we see. So when more channels open, we get a greater change in the membrane potential. And this is why a greater frequency of action potentials is going to lead to a greater postsynaptic response. Because the more action potentials we fire, the more neurotransmitter release. The more neurotransmitter we release, the more receptors we can activate, and the more active receptors, the greater the response. Whether you have excitatory or inhibitory conductance, that's going to determine whether or not the neuron then fires another action potential. So the excitatory postsynaptic potentials, or the EPSPs, are going to depolarize the cell. That's caused by sodium channels, which reverse at roughly positive 56 millivolts, or non-specific cation channels, which reverse at 0 millivolts. Both of those are sufficient to depolarize the neuron above threshold. Inhibitory postsynaptic potentials are going to either hyperpolarize the membrane potential or cause shunting inhibition. Inhibitory postsynaptic potentials are caused by potassium or chloride channels, right? Because both of those reverse at negative potentials, negative 100 and negative 78 or so. So when you get a variety of different inputs in your dendrites, all of those funnel into the cell body, sum together, and then determine whether or not the target cell will fire an action potential. Okay, that about does it for synaptic signaling. Email me any questions that you have on the material, and I look forward to seeing you for Lecture 6.